I'm Kate Murray, the President and CEO for Canada's Stem Cell Network, and I'm pleased to be here on behalf of my colleague, John Draper, our Vice President of Research and Training. John is off sick today, so I will do my best to fill in for him as your moderator. This is an introduction to biological GMP manufacturing, preparing for early phase clinical trials. And we are presenting this today on behalf of our friends and our partners at BioCanRx and the National Research Council. This is our first webinar in the series. I believe there are 3, and the Stem Cell Network is happy to host today's session. The next 2 sessions will be hosted by one of our other partners. While we meet today in a virtual platform, the work of Canada's Stem Cell Network occurs from coast to coast on the traditional and ancestral and unceded territories of many Indigenous peoples and nations, including the Anishinaabe-Algonquin Nation, where SCN's offices and headquarters are located. And we honor and recognize and respect our Indigenous nations as the traditional stewards of the lands and the waters where we work, meet, and play. So Stem Cell Network, as many may know, is a Canadian not-for-profit that supports 3 main objectives: research in the regenerative medicine space, training the next generation of talent, and supporting the knowledge mobilization and transfer of research outputs for public goods. We were formed more than 20 years ago and continue to be supported by the Government of Canada. To date, we've invested nearly $150 million into our 3 programs: research, training, and knowledge mobilization. And many of our activities we do in collaboration with partners such as BioCanRx and the NRC. We are an organization that is designed by researchers for researchers, and we bring together scientists and entrepreneurs and policymakers and clinicians to help further the competitiveness of regenerative medicine globally. And we hope to continue doing this for many years. BioCanRx has been one of our sister networks for many years and has been a wonderful partner to work with and to do training with. And we are very excited to be working with BioCan and now this new partner with the NRC on today's series. So first, before we move into all of the wonderful talks, we need to learn a little bit more about who's joining us and in the audience today. I'm going to turn on a poll that has 3 questions, and it'll pop up on your screen. Hopefully it won't take you more than about 20 seconds to answer. And if you could do that, that would be great. And I believe we will also display the results so you can see who we've got in the audience this afternoon or this morning. So go for that. Okay. Do my polling folks have the results to share? Or do we still need some time?
Still collecting, sorry.
Oh, we're still collecting. So maybe as we continue to collect those polling responses, I'll just go over a few housekeeping notes for today's webinar. There will be times where we'll ask for audience questions. You can submit these through the Q&A chat box at the bottom of your screen. The session will be recorded and posted publicly on the Stem Cell Network's YouTube channel in case you'd like to revisit it or share today's content with others. Closed captioning is enabled for this session, and you can choose to show or hide the automated closed captioning. And after each session, a post-event survey will pop up. If you're planning to attend all 3 of the sessions that we're doing, you just need to fill that post-event survey at once. This really helps us to understand what was working with these sessions and where we can improve, and how to think about offering this down the road. So are we ready with our polling yet, Gus? We are. Okay. So a lot of people from academia, some from government, private sector, and other. Oh, good. We see lots of confidence there with folks in wanting to bring their product to clinical to GMP, and some are neutral. Yeah, that makes sense. And what kind of biologics are you developing? Most people are in the cell and gene therapy space. So terrific. Well, thank you for sharing this information, and this will help our speakers too as they gauge their remarks for today's session. And so finally, I'd like to offer a big thank you to the speakers for participating in this series, particularly today's speakers, Sanjana Fridman, Marie-Michelle Genois, Brian Lichty, Gail Payette, and Jennifer Queasy, as well as the teams at SCN, BioCanRx, and NRC for their leadership and commitment in bringing together today's event. These take a lot of planning and effort behind the scenes before we actually get to this moment, and we couldn't do it without our teams. So our first speaker of the series is Dr. Brian Lichty, who will be giving an overview of the clinical translation pipeline, and he'll give some examples from his own expertise in oncolytic viruses and related immunotherapies. He's been a professor in the Department of Pathology and Molecular Medicine at McMaster, at the McMaster Immunology Research Center since 2004. He's also a co-founder of Turnstone Biologics. Many of you will be familiar with Turnstone and their great success, and the director of the Robert E. Fitzreney Vector Lab at McMaster, where clinical grade viral vaccines are manufactured for human clinical trials. He's received his Ph.D. from the University of Toronto and holds a bachelor's from the University of Guelph. So on that, I will turn the floor over to Brian.
Hi, can everyone hear me? Is that working?
Sounds good, Brian.
Okay, and I will share my screen. Is that working?
Yes, working well. We're now in presentation mode.
Okay, so hello everyone. I hope I can make this relevant to most of you. I see that many of the participants are really, about taking cell-based and gene therapy biologics to the clinic, which isn't really what I do. Everything I do is based on viruses, but I think that applies to a large degree, and some of these things are generic across biologics and GMP manufacturing generally. And I assume that in many of the cell-based therapies, there's an element of using a DNA plasmid or vector or something to modify your cells. So some of this will apply regardless of what actually is the thing you take into the clinic. So this is sort of the overview map, I guess, roadmap that I share with people who ask me about taking, in this case particularly, let's say an oncolytic virus or a viral vectored vaccine to the clinic? What are the things you need to do, and how do you string it together, and what are all the considerations? There's a bunch of steps that apply in this case, and I think they apply in many of the other biologics that any of you are thinking about. In broad strokes, of course, you've got to have your non-clinical studies. That's usually work you've done in your research lab in a research animal model, typically a mouse, but it varies. The point of those studies is to demonstrate that you've got a lead candidate, whatever it is, that can provide potential benefit to patients or volunteers in the case of a vaccine. And that is opposed to the tox studies that you'll also have to figure out how to do. And that's more specific, I think, to whatever it is you're developing. But, you know, the two things that can get you into the clinic are potential benefit and demonstration that there's a relatively balanced limited risk for whatever you're developing. Selection of your lead candidate is very important. A lot of research labs compare and contrast and play around with a variety of potential candidates and get to a point where they think, okay, this is the best thing, and that's what I want to take forward. How you get to that is important, and what you do at that point is very important, because that will determine how ready you are to talk to somebody about manufacturing, and do you have your ducks in a row so that the next steps can go smoothly. And then there's a bunch of issues around manufacturing. If you're making cells, you may not need to think about the cell line you're going to use, but for everything else we do, we tend to make things in cells, whether they're proteins or viruses. And even if you're making cells, you may be making a lentivirus or something like that to modify your cells. So cell line selection crops up there as well. And then there's, how do you take the way you make whatever it is you want to take to the clinic into the clinic and manufacture it along the way. There's always some degree of process development. So that's the development of a series of processes that allow you to, in a GMP fashion, manufacture the biologic that you then can get approval to put into human subjects. And it really matters whether that's a totally new process for the facility you're engaging with, or if they're able to simply adapt or fine tune in some sort of existing process. And I'll get to that. So that's broad strokes, how this all works. So you start by selecting your lead candidate. And that selection needs to be well justified. I'll mostly speak to viruses here, but this could be, the lentivirus that's going to modify your cells, for example. So choices have to be made about that as well, that are, this is relevant too. So how novel is it is a big question, right? So is this something that's never been in humans before, or is this a variation on what's been in humans before? And so whether or not there's a clinical track record for the technology and you're doing something similar to but different makes a big difference. If you're expressing a transgene, why that transgene and not something else, and is it safe? And you have to justify the target. So your non-clinical work needs to justify what you're doing, so that you can convince regulatory bodies and clinicians and patients that there's some chance that there's going to be benefit to what you're doing. The key bit here is, once you've done all of that, the actual lead candidate, if it's a viral vector particularly, has to be very well characterized, and you need a clonal source to create a seed stock that ideally is generated and isolated in a GMP compliant manner. So Unfortunately, a lot of academic research labs, because of how they function, they have a team of people involved that generate reagents and candidates in not an entirely ad hoc fashion, but individual trainees or postdocs or whoever may generate different candidates, and then those are compared, and ultimately you make a choice. But the history for each of those things is really important. You need to be able to either redo the generation of that candidate, and then you sometimes may need to redo some of your non-clinical work with the actual candidate, because work done with other things, in some sense, preclinically don't count because it's not the actual clinical candidate. So that's just something to keep in mind as you're moving towards the clinic. You want to pay really close attention to how you generate the materials you work with, the documentation around where they came from, the reagents used, lot numbers for everything that goes into it. And if you're getting towards the clinic and you're serious about going to the clinic, you should look into using reagents that are somewhat or completely GMP compliant, so that the thing you hand over to the manufacturer is easy for them to work with and move forward. Very briefly, clinical trial design matters, and the sooner you have an idea of what your clinical trial is going to look like, the easier it is to figure out how you're going to biomanufacture. So there are different sort of guidelines and rules about what testing you have to do depending on, say, route of administration. There's a big difference between IM and inhaled and IV. And I've manufactured things for all each of those routes, and there are differences in terms of the testing you have to do to be compliant. Of course, you have to think about dose. So if you've only worked in, say, mice, how much material do you think you'll need to treat humans? You can scale it, and there are accepted ways to do that, which maybe are not entirely predictable for a biologic, but it's the best we can do. And you may have data from another species in terms of your, say, tox testing and so forth. And then formulation of format and storage are all really important. And you maybe can't do what you've been doing in your research lab in terms of, what do you put your, say, virus in to store it and freeze it, and the container you keep it in and so forth may not be GMP compliant. And you have to make sure that you're working towards or thinking about ways of doing these things that will be GMP compliant by the time you get there. Knowing the scale of your phase 1 or phase 1-2, and what is the dose and what is the route, help you determine what sort of scale and concentration of material you're going to need. If in this case, the virus that I'm speaking to primarily is the actual product. If it's a vaccine, it goes IM, it'll be in this format, this concentration, this volume. But if you're going to go IV or intranasally or inhaled, these things may change. So you got to think a lot about what you're planning on doing. And the sooner you can kind of map out with your clinician partners and whoever else is going to be involved, and maybe even speaking to regulatory and a pre-CTA, what you're planning, it'll help you think about, what manufacturing is going to work best for your scenario. I'm going to speak a little bit to cell line selection. So when we manufacture viruses, or people make viruses to use to modify cells, these are made in cells, and hopefully you can use one of what I would call the usual suspects. So if it's for monoclonals, it could be CHO or HEC293 or Percy6, or if you're going to make an influenza, it might be an MDCK. oncolytic viruses could be made in HeLAs or A549s, or there's certain other vaccines that have been made in these cells. Maybe you want to make recombinant proteins in insect cells or bacterial cells. In any case, you're going to have to pick a cell type to produce your biologic in. And in some cases, the manufacturing facility you go to may not actually have their own stock of those cells that you can use. In some cases, you got to bring the cells and the thing you want to manufacture in them. So be aware of that and think about what cells you're going to want or need to use. And if you're going to source cells yourself, be careful. You can't necessarily just use whatever you've been using in your research lab all along, because provenance is very important. You have to know in great detail, where did the cells come from? What have they been exposed to, especially animal products? What is their passage number, and so on and so forth. In many cases, you have to go out and resource cells, even if it's the same cell line you've been using all along, to make sure you can be compliant in this regard. And ideally, you want to get to a situation where there's no animal products, no serum, no animal enzymes being or have been used. And in many cases, to enable large scale production, suspension adapted is ideal, but it doesn't always work. And so, there's ways to grow all these cells in an adherent format so that you can manufacture different viral products or recombinant proteins. But selecting the cell line, knowing where it came from, and in many cases, not simply trying to use your research grade materials that you've been using for all your non-clinical work is going to be necessary.
We just asked that everybody put themselves on mute. Martin, you're not on mute.
Ouais, ça sera pas très long. Je veux juste te dire que j'ai eu l'approbation pour engager Suzanne pour 1 an.
Sorry about that, Brian. Thank you, Martin.
Oh, okay. And then, manufacturing, obviously you need to have a scalable process. And what I mean by that is very often a process that's used in a research lab isn't very scalable. So for example, if it involves centrifugation to collect cells or virus or what have you, that's not very scalable. And, we need to get away from things like that as we go to larger and larger scale. So, and then the culture conditions might not be terribly scalable either. So, ideally, you've already started working on identifying the process, and you have something that is amenable to scaling that you can tech transfer to the facility you engage with to do the manufacturing, or you engage with a facility that already makes whatever it is that you want to make. So if they already make monoclonal antibodies, that obviously is great news, or they make adenovirus or AAV, or they, make lenti-transduced cells. Then tech transfer gets a lot easier, and you can sort of plug into whatever it is that they are currently doing for others. But there still will need to be some degree of process development, which will be done at a small scale. There may be upstream process development, depending on any changes to, say, producer cells, or if there's an infection or transfection step. That will have to be tested to see if it's compatible with their existing process. And then there's downstream process development to purify your product to achieve a desired purity and potency. And then you have to think about formulation and storage, and maybe somebody's already figured this out for whatever you're doing, but maybe not. And whatever you're doing in your research lab may or may not be GMP and clinically compatible. And along the way, there's certain steps. So if you're making, say, a viral vectored vaccine or a gene therapy, a viral vector gene therapy product, for example, you need to think about the cells you manufacture them in, like I said, and then you may need to make your own cell bank, which then is thoroughly release tested so that it's, you know, a GMP compliant reagent to be used to make your viral vector. And then you're going to want a viral seed bank, and then a master seed bank or master virus bank, that you use with your cells to make your actual clinical product. But all of this needs to be worked out with your whoever you're manufacturing with, and the more you sort of sort out early in your own lab and with them, the easier it'll go when you actually pull the trigger. One thing I'll speak to very briefly that what didn't used to be a thing, but it now is, I would say, unfortunately, is that biologics generally need to undergo an environmental assessment. So we ran into this with a viral vectored COVID vaccine, a couple of them that we manufactured at McMaster and took into the clinic during the pandemic. And we had not run into this before, but my understanding is that any biologics that are brought to Health Canada are also referred to the environmental assessment folks, and you need to go through this process to convince that whatever it is you're going to release into nature, meaning that you're going to put into volunteers or patients, is not going to have an environmental impact on the country. And so you'll get asked questions like, will your T cells affect the fisheries and our forests and so forth? And it's kind of hard to find publications that back up your statements, but it's the best you can do. So that's an unusual and kind of weird experience that we've been going through lately, and I would recommend simply finding a consultant who deals with this and engage them and help you through the process. Another thing you'll have to think about is assay development. So there's going to be a whole bunch of release tests that have to be performed on whatever it is your biologic before you can put it into people. And this varies a lot, so it's hard to speak to this generically, but in many cases, we outsource these tests to a third-party service provider. You have to figure out, usually in consultation with Health Canada, which ones you need to include, and therefore which ones might need to be developed and then validated, because some things will be very specific to your biologic and not just generically available from, say, BioReliance or Wuxi Aptec or these companies that do this work for us. Some assays you will have to develop yourself, and the sooner you get working on that with your manufacturing partner, the better. For example, what is your potency assay, and how do you measure what is actually a dose? And if you need reagents to do some of these assays, you have to think hard about, how do you source them, and will you end up, meeting GMP compliance? So, for example, one of the big challenges in the oncolytic virus world is that you have to test for adventitious agents, meaning anything that could be in your product that you don't even know about, that could be a pathogen because you grew it in cells. But if your drug kills the reporter cells, you can't detect other pathogens. And so you have to be able to completely neutralize your virus with, say, an antibody or antibodies. And is this even possible? And how do you source those antibodies and validate the assay? There are some things that you may run into that certainly were surprises for us when we first did, that I'm sure you've never thought of before. So the sooner you become familiar with some of these assays that you may need to do and figure out how to tackle them, the better off you'll be down the road. In terms of tox testing, I won't really get into this. we've done a mixture of, in the past, formal or informal, sort of GLP-like tox testing with the viral vectors and vaccines we've developed. But importantly, for your tox testing, whatever it is, ideally it should be some sort of engineering run or tox slot. The whatever goes into the tox testing ideally is produced using the clinical process that you're going to use to make what you take into the clinic. And so this can create a timing delay. So once you've developed a process and you have the ability to make, a small manufacturing run using that process and can make material for some tox testing, you have to decide whether or not you delay there and wait for the data from the tox testing before you pull the trigger on manufacturing at a larger scale, or whether you can't deal with the timelines and you're confident about what's going to come out of your tox testing, and then you just plow ahead. And of course, there's a big difference in how academia and industry deals with these sorts of decision points, but the interaction between tox testing and manufacturing is important in terms of decision points, which is why I bring it up here. And then the other thing is, if you manufacture things differently for clinical work and use that sort of material for tox testing, you might want to redo some of your non-clinical or preclinical efficacy testing just to make sure you haven't, by changing how you manufacture things, lost the efficacy that you were seeing using your research grade manufacturing, depending what you're working on. And so this is just sort of a timeline that I share with folks who want to take, say, an oncolytic virus or a gene therapy vector or a vaccine, a viral vector vaccine, into the clinic, and the different steps that you have to go through in terms of picking your lead, performing non-clinical studies specifically with your lead. Even if you've done a bunch of work to pick the lead, you want to have solid data for that lead. And you have to isolate it or regenerate it in some cases in a way that you can hand that off to manufacturing, and it'll be GMP compliant, and it's really easy to figure out where everything came from and what it was ever exposed to and how everything was done, and it's well documented. And if you need to pick a cell line, the sooner you do that, the better. And if you need your own cell bank, that's going to take time, and then that cell bank's going to have to be release tested. There's a lot of testing that you would apply to a cell bank to prove that it's a GMP compliant reagent for producing a vaccine, say. And then, and while you're doing that, you'll want to be doing process development with whoever's going to do your manufacturing. That'll eventually lead to your tox slot and a master virus bank in this case, which can then be taken into clinical production and release testing. Maybe while you're doing your tox testing and finishing off everything you need for your CTA. And I'll stop there.
That's great. Thanks, Brian. That was a lot of great content in a very short period of time. I already saw one Q&A, one question come up in the text box. We will get to questions. We'll do that at about 5 after 1, after we've just had our second presentation today. And I'm really pleased to be able to introduce to you Sanjana Fridman, who's a regulatory and compliance and enforcement advisor at Health Canada. And she's with the Drug GMP HQ Team 3 in the Health Product Inspection and Licensing Division. Sanjana, can you really get that all on a business card? She joined Health Canada in 2018. Prior to that, Sanjana worked in industry in the area of sterile injectable products. She holds a master's in radiation oncology. And Sanjana is joined by her colleague, Dr. Marie-Michelle Genois, who is acting senior biotherapeutics quality reviewer. at the Biologic and Radiopharmaceutical Drugs Directorate at Health Canada. She's joined in 2021, and prior to that completed a postdoctoral fellowship at Harvard Medical School in the area of cancer biology. She holds a Ph.D. in cellular and molecular biology. Two highly accomplished women, and really pleased to have you both join us on behalf of Health Canada today. And I'll turn the floor over to you.
So hi, I'm waiting for the PowerPoint slide.
All right, let me just pull that up. It's loading. Okay. Thank you for the very kind introduction. Give me a second. Here we go. Let's share our screen. Let me know when you can see the screen.
I can see it. Yeah, thanks.
Perfect.
Okay, so yeah, like my name is Marie-Michelle Genois. I'm a quality reviewer at Health Canada, and yeah, I'm today with my colleague Sanjana.
Hi, my name is Sanjana Fridman, and I am a Senior Regulatory Compliance and Enforcement Advisor with the GMP Inspection Program, and yes, that is a mouthful.
Yeah, we would like to thank the organizer for inviting us today at this webinar, and we are very happy to discuss on how to prepare a quality package for an early phase clinical trial. So we would like to make sure that the opinion expressed in this presentation are those of the presenters. And now we will turn off our camera to reduce the distraction. Okay. So the focus of today's presentation is on elements that a new investigator should have in place to support the transition to a phase 1 clinical trial, and the requirements needed prior to securing a contract with the manufacturer. From a quality point of view, the main consideration for phase 1 is the safety of the product. This presentation is divided into 2 parts. The first part will be about, regarding the CT review process and the quality requirement. And the second part, my colleague Sanjana will discuss about the GMP regulatory framework for Biologic, and she will present a GMP case study. So let's start with some statements that you might have heard before, but which is not necessarily true regarding clinical trial. So the first one is obtaining a CTA authorization takes a long time. In reality, 30 days is the maximum days before Health Canada notifies the sponsor if there is no objection. Another one is that you need a DEL, a drug establishment license, to be GMP compliant and to manufacture clinical drugs. In reality, no DEL is needed, but clinical drug for human use must be manufactured, handled, and stored in compliance with GMP. Then all documents must be provided in English and French. In reality, either language is accepted. And the last one, there is no way to ask question advice to Health Canada prior submitting a CTA. In reality, sponsors are encouraged to request a pre-CTA meeting with us to ask for advice and question, and these meetings are free. So what is the review process for clinical trial application in Canada? So once you submit your CTA to us, it will be screened for completeness, and if deficiencies are identified, you will receive a request for clarification, and you have 2 days to provide the information requested. The clock will start once the CTA is complete, and 30 days is the service standard. Then the review is performed by quality and clinical teams in parallel. The sponsor is responsible for resolving any issues identified by the reviewers within 2 days. If the CTA is deemed acceptable, a no objection letter will be issued. If during the course of the review, the sponsor is unable to provide the requested information or there is an objection from the review team, the sponsor will be notified with some option. And now, what do you need to include in your Biologic quality package? 3 types of documents are expected to be submitted in your application. The first one is the study protocol, which should describe the objective and methodology of the clinical trial. We expect that the maximum proposed dose and the information related to the target population is included in this document because the dose and the patient weight are important information for impurities calculation. Another document expected is the investigator's brochure, where the safety, non-clinical, and clinical data are described. The quality information expected in this document is the physical, chemical, and pharmaceutical properties and the formulation of the drug. And finally, the chemistry and manufacturing information should be provided. This includes all the quality information related to the drug, and it is separated into parts, the drug substance and the drug product. Each part are divided into sections, including the manufacturer, the characterization, the control, the reference standard, the pharmaceutical development, the stability, and adventitious agent safety evaluation. So in the next slides, I will go through the different sections and describe what are the expectations for a phase 1 CTA. But first, I will give you a general picture of what should be implemented at the beginning. So in early phases, the emphasis is made on producing a safe and accurate dose to the participant. The focus is made on raw material, virus safety, endotoxin, bioburden, and impurity clearance. The control strategy will be mainly based on general knowledge of therapeutic protein, for example, and on limited manufacturing experience. As the sponsor moves forward in development, the control strategy will evolve to reflect the accumulated product knowledge to ensure safety, efficacy, and consistency. For example, at the later stages, the specifications are refined based on manufacturing experience, a stability program, and a robust potency assay should be implemented. So now let's talk about the quality information that you need to provide. The first section I will discuss is the manufacture of the drug substance. A detailed description of the manufacturing process, including a flow chart, the scale, and the manufacturing site should be included. In-process controls that are in place to monitor endotoxin, bioburden, mycoplasma, and virus in the harvest material should be clearly indicated. It is also important to provide data to demonstrate that the cell banks have been characterized to make sure that the cells used to produce the drug substance are really what we expect to be, that they do not contain any contaminant, and they can consistently produce the intended product over storage. At early phases, a one-tier system is acceptable, but as the product development continues, a two-tier system is expected for consistency. Control of raw materials is also important because it represents a source of potential contamination. The quality of non-biological origin material should be compendial. If there is no compliance standard for the material, it should be controlled with appropriate in-house specification. For human and animal-derived raw material, as bovine serum or porcine trypsin, it should be avoided as they represent a potential source of viral contamination. When it's not possible, the material should be supported by appropriate documentation. Evidence could be an official document from the manufacturer, such as a certificate of suitability. A table as like this one can be helpful for the reviewer. So now the next piece of information required is the characterization of the product. A summary of the general properties like the primary structure or the biological activity should be described in this section. It is expected that the characterization of the molecule will evolve over the course of the development. So when a change is implemented, like example, you want to use a different manufacturing process, comparability assessment needs to be provided to demonstrate that the lots are comparable to each other in terms of product quality. Comparability studies are generally based on batch analysis, characterization, and stability data. Potential contamina and impurities should also be described in this section. It is understood that impurities may not be fully characterized at early stages, but they should be monitored during the process, controlled at release, or supported with a safety assessment to ensure that they are unlikely to pose a safety risk to the participants. So what is an appropriate safety assessment? It is an assessment that can include clearance data, platform knowledge, and a worst-case scenario calculation to support that the residual levels are safe based on permitted daily exposure limit from literature or in-house tox studies. So here are some examples. You can see on the left that in the in-process bulk material, bioburden, mycoplasma, and viruses should be tested for every batch manufactured. In the middle box are represented the impurities usually tested during the process or at release. Note that endotoxin and bioburden are key impurities that should be monitored throughout the process. And finally, on the right are the ones that can be supported with a safety assessment. As example, the residual solvent like DMSO. So note that a change to the cell line in scale or to the purification columns will have an impact of clearance of those impurities. So let's discuss now about the controls. In the control section, the specification used at release, testing, and batch analysis results are provided. In early phases, specification can be wider for some attributes due to limited number of batch manufacturer and variability of the process. Specification limit for appearance, identity, protein concentration, purity, safety, and potency are expected. The specification should be reflective of the manufacturing experience with tox batches and evolve with the experience from the clinical batches. At least one batch analysis data manufactured using the proposed process should be provided for drug substance and drug product. So in the event of an out of specification, the batch cannot be used for clinical use until you notify Health Canada and receive a no objection letter. Supporting data and a justification will be required to demonstrate that the out of specification observed do not impact the product quality and patient safety. So at early phases, we require mainly a representative batch to be the interim reference standard. It needs to be clear which reference standard is being used, and a release and characterization result should be provided. A reference standard needs to be defined at later phases since it will be used to validate analytical methods. So now, how can we better understand the product behavior, the trends, and degradation pathways over storage? So it's by performing stability studies for both drug substance and drug product. So there are three types of stability studies. The real-time studies that are performed in the actual storage condition, while the accelerated and stress studies are performed at different conditions to enhance product degradation. When appropriate, in-use stabilities covering the preparation and the administration time should be performed. Typically, 8 hours at room temperature or 24 hours at 2 to 8 is allowed with supporting data. The in-use shelf life proposed should be a balance between the potential risk and the practical requirements for conducting the trial. Method able to demonstrate product stability and that can detect changes, impurities, and potency should be included in the stability program. Clear stability specification for stability indicating attributes should be stated. And also, the container used for stability studies should be representative of the actual container closure system of the product. And finally, a shelf life should be proposed based on the stability data. The maximum shelf life should not exceed 2 times of available data or no more than 12 months. So for example, if you provide 9 months of real-time stability data, you can propose a maximum of 18 months shelf life. If you provide 18 months of real-time stability data, you can propose a maximum of 30 months shelf life. So any shelf life extension should be notified to Health Canada along with supporting data. Now we'll go over some specific section for the drug product. In some cases, the drug product is in direct contact to new components during the preparation and the administration, and this material can affect the product quality. So compatibility studies need to be performed to demonstrate that the product quality and safety are maintained throughout the preparation and administration. For example, the drug product might be not stable in the bag with the diluent during IV administration, and the product degradation can cause particle accumulation and a decrease in protein concentration. So the compatibility study should include testing for appearance, protein concentration, potency, and purity. A brief description of the manufacturing process and control need to be provided with a flowchart and a batch scale. It is expected that bioburden, pre- and post-testing filter integrity are included in the in-process control. Regarding the control strategy for the drug product, additional specification compared to the drug substance specification should be in place to assure that the product quality and patient safety. This leads us to the last section where you need to provide the adventitious agent safety evaluation. To mitigate the risk of contamination, the cell banks and the unprocessed bulk harvest need to be tested for retrovirus, sterility, and mycoplasma. In vitro and vivo assays and antibody production tests should be performed for the cell bank testing. Alternative molecular virus detection methods like next-generation sequencing may be used to replace the viral assays. When applicable, virus clearance studies using relevant virus spike preparation must be performed to validate manufacturing steps that are designed for viral removal. The retroviral safety margin should be considered the worst case condition, considering the maximum dose and the highest of virus-like particle in the unprocessed bulk harvest. A small-scale study can be performed, but should be representative of the actual manufacturing process. So now we have been through all the section of the chemistry and manufacturing information. I would like to provide some reference to help you to prepare your CTA application. So the ICH guidelines should be taken into consideration when preparing the quality component of the CTA application. And below here are the Health Canada guidance to help you prepare your package, file, and manage your CTA. So this is the end of the first part. My colleague Sanjana will continue the presentation and discuss about the GMP regulatory framework.
Thank you, Marie-Michelle, for all that. So next, I will briefly discuss GMP and their application in early phase trials. I'll start with a brief overview of the regulatory framework for biologics. These are subject to the Food and Drug Regulations Part C. Part C is further divided into divisions, each with their own focus. 6 of them apply to biologic products, and we're going to focus on 2 today. Division 5 for clinical trials, and Division 2, which are the GMPs. Part C, Division 5 of the regulations govern clinical trials. All clinical trials must comply with Division 5. One requirement of Division 5 is for the study sponsor to ensure that all clinical trial drugs comply with Division 2. Or to put it more simply, they must be GMP compliant. But what is GMP exactly? GMP stands for Good Manufacturing Practices. They are a set of internationally recognized standards that apply to drug manufacturing. They ensure that the drug products that finally reach patients are safe, effective, and of acceptable quality. In Canada, you can find them in Part C Division 2 of the Food and Drug Regulations and in Health Canada's GMP Guide for Drug Products, Guide 1. And you should also be aware of resources from international committees such as ICH and PICS, as they also publish international guidance on specific topics. So if you look here to the chart on the right, this is what the GMPs in Canada look like. And if you think they look intimidating, remember that they were written to be applied to market authorized products. However, per Division 5, clinical trial drugs are still required to comply with GMP. Now, this can be challenging in early phase studies. At this point, the product is not yet fully developed, the manufacturing process is in its early stages, and then there's the added complexity both of manufacturing for clinical trials and of manufacturing in biologics. So acknowledging all of this, we have to recognize that the entire scope of GMP cannot be applied at this phase of development. There must be flexibility in the application of GMP because the requisite data are still being established and refined. So with all this in mind, how do you comply with GMP when producing for a phase 1 clinical trial? This brings us to our case study, where we will be exploring the journey of a fictional biomanufactured product transitioning from an R&D to a GMP setting. The goal of this exercise is to identify and brainstorm at a very high level some key areas where you can be proactive when it comes to meeting GMP requirements. So let's say you're a PI developing a cell-based therapy that has shown promising results in preclinical studies. You've been producing this in the lab using a process flow that looks something like this. You've established a parental host cell and an expression construct containing your gene of interest. You've transferred the expression construct to your host cells and selected for the transformed or transfected cells, and you've established expression construct stability in your propagated recombinant cells and successfully expanded them into a cell bank. So now that you've achieved all of this, you want to bring your product to clinical trial. What do you do next? So first, let's consider the big picture and imagine how this fictional process may evolve to full-scale GMP manufacturing using a two-tiered cell bank system. The first step in manufacturing would be to expand your research bank cells into a master cell bank. And then by expanding one or more vials of your master, you would then generate your working cell bank. The working cell bank is what you use to expand and yield a single lot of your end of production cells, which is the final product. This final product will then be filled, packaged, and transported to the patient, and so on. So this is the type of process that would be the end goal of clinical and commercial development. But for an early phase clinical study, as Marie-Michelle had mentioned earlier, a two-tiered cell bank is not expected and a single cell bank would be sufficient. So let's simplify the process diagram to resemble more closely what early phase manufacturing might look like. So up until the generation of your research cell bank, all of your activities are considered research and development and not manufacturing. However, the GMP cell bank is considered the starting raw material for manufacturing, as this is what's used to produce the final product. From this point onward, we consider manufacturing to have started, and at this point onward, GMP must be followed. So when it comes to navigating the transition to GMP, the best way is to integrate GMP principles as early as possible. But how do you do that? First, remember that the objective of GMP is to control product quality by establishing a consistent and controllable process. At its core, if you can control the quality of your process inputs and you can control your process, then you control the output of your process. That is to say, you can consistently manufacture a final product of acceptable quality. At this phase, the GMP information you'll have to provide will fall into three main buckets: information about your process inputs, which are your starting materials, about your process controls, and the analytical methods that you'll use throughout. Recall that as the trial sponsor, part of the information required from you in the CTA is to support manufacturing consistency, which is the goal of GMP. So integrating GMP principles early on in development will help you prepare your application as well. So let's start with our process inputs. These are our starting materials. Remember, what is the goal of GMP? Manufacturing consistency. And how do you achieve consistency? By limiting variability. So how might we limit variabilities in our starting materials? Well, the first step is to identify them. For a phase 1 trial, your starting material would be your cell bank, as well as any ancillary materials. These are components that may not appear in your final product, but these still represent process inputs that can potentially introduce variability. These are things like your culture media, buffers, growth factors, and so on and so forth. So now that we've identified all our starting materials, how do we limit variability, and how do we ensure that they are as uniform as possible? GMP formalizes these questions into an exercise called qualification. Qualification is the process of acquiring and evaluating data to establish the overall suitability of a specific component to ensure quality. In phase 1 studies, this will focus on ensuring safety, as the product's safety is not yet fully understood. Overall, the goal of qualification is to be able to address three main questions. Number 1, have you holistically assessed all your starting materials for their characteristics and their impact upon the process and final product? Number 2, how will you confirm that your starting materials possess the properties that will provide the desired outcome in your process? And finally, number 3, how will you confirm the identity and quality of your starting materials before you use them in manufacturing? So with all this in mind, let's try qualify our starting materials together. Starting with our cell bank, how might we evaluate the quality and consistency of our cell bank? So first, we will have to consider how the cell bank was generated. This will encompass activities that were done in research and development, such as how did you select and prepare your host cell and expression construct? How did you generate your recombinant cells? Did you follow a standardized procedure? And what specifications and acceptance criteria will you be using to assess the quality of your research bank cells before releasing them for use in manufacturing? And a hint here, you might notice some significant overlap between these parameters that you have to consider when qualifying your materials and those that are required to be provided as part of your CTA. So moving on, how might we qualify our ancillary materials? Well, first of all, can you identify all of them? Do you understand their purpose in your process? Where will you source these materials from, and do you have any alternative suppliers? Next, on the subject of sourcing, have you qualified any of your suppliers? Vendor qualification is an exercise meant to establish confidence that your vendors of choice are reliable suppliers. Qualifying a vendor entails exercising due diligence upfront. For example, have you audited your vendor's manufacturing facility? Have you checked for their familiarity with key principles such as validation, cleaning and sterilization, or cross contamination prevention? Do you have a quality agreement with them that clearly defines roles, responsibilities, and expectations for product quality? All of these due diligence activities build confidence in your selected vendors, and they ensure that you are receiving materials of adequate and consistent quality. This due diligence should be taken every time you engage a new supplier, and this is especially important when sourcing materials where GMP certified facilities may not exist. For example, in Canada, clinical trial drugs are exempt from requiring a drug establishment license. But by having a qualification program in place, you can confidently assess the GMP compliance of any third parties you may choose to partner with. So, moving on, finally, back to our ancillary materials, what specifications and acceptance criteria will you be using to ensure that they are suitable for use prior to being released to production? Remember, the goal of all of these considerations is to ensure, with data, that your starting materials are as consistent in their safety and quality as possible. So let's move on to the manufacturing process now. Remember, at this phase, what is the goal of GMP? Manufacturing consistency. And how do you achieve consistency? By limiting variability. So how might we limit variability when the process is not yet fully validated? Well, at this phase, the process should still have well-defined go or no-go decision criteria for key steps. These decision criteria are a way to monitor your process. They ensure that the in-process material at key steps is of acceptable quality, quantity, and yield. They also allow you to gather data as development proceeds and to assess the impact of any changes or excursions to your process. So when it comes to identifying potential process controls, what might you use to monitor? Well, for a product like this, indicators of cellular growth, such as the ones listed here, are all measurable attributes that give you data about whether your production is proceeding at the expected rate or if it's occurring too quickly or too slowly, which may indicate that there's a problem. Monitoring for things like sterility, bioburden, or impurities can also quickly alert you to any failures that would require a prompt investigation. Now, this is not an exhaustive list by any means, but it should give you an idea of the type of controls that can be considered. So now that we've had a quick look at some possible in-process controls and we've considered how to control the quality of our starting materials, you may notice they all have one thing in common. They rely on testing. Which leads us into our next consideration, which is analytical testing. Remember, at this stage, the process is not yet validated. When transitioning over to GMP production, you need to be able to rely on accurate and consistent testing in order to assess if your process is in control and whether the final product conforms to its specifications. So it is critical that you have tests to evaluate your starting and in-process materials as well as your final product. Remember, at this stage, what is the goal of GMP? Manufacturing consistency. And how do you achieve consistency? By limiting variability. So now let's consider how your tests can contribute to variability. The two main questions to answer here are, what tests will you need, and are they accurate and reliable? So when it comes to the type of tests you'll need, think about what you'll be measuring. First, you'll need tests to measure any critical quality attributes that can be used as an in-process control, as we discussed earlier. You'll also need tests indicative of product safety and quality, such as a dose-defining assay and potency and so on. These are important to monitor both throughout your process and when testing your final product. Next, how do you ensure that your test methods are accurate and reliable? Some questions to ask here include, Are your analytical methods specific to your product? Do they have acceptance criteria that are appropriate and supported by preclinical data? Again, you might notice some overlap with the methods that will have to be submitted with your CTA. Are your methods compendial, or did you develop them in-house? Is the method suitable for its intended purpose? And are there any appropriate reference standards or reference materials available? These may not be required for early phase studies, but they will assist in the validation of your methods in later phases of development. So now that we've considered our analytical methods, we've arrived at the end of our process and at the end of our case study. We've looked at some strategies to control the quality of our inputs and control our process in order to consistently manufacture a final product of acceptable quality. First, we've analyzed the quality of our starting materials by qualifying them. We've thought about some in-process controls to monitor manufacturing at key steps. And we've considered our analytical methods, which will anchor the entire process and product quality before the process is fully validated. And by addressing all of these categories, we've positioned ourselves ideally to meet the goal of GMP at this phase, which is manufacturing consistency. So here I've compiled a list of all the technical references used for today's presentation for your reference. But in conclusion, overall, first, the primary objective of an early phase clinical trial is to ensure the safety of trial participants. GMP requirements will increase in stringency as you proceed through development, but as the sponsor, you should always follow GMP principles. Second, by controlling the quality of your starting materials and your process, you can consistently manufacture a product of acceptable quality. And finally, by proactively applying your understanding of product and process characteristics, you can identify and limit sources of variability.
So we would like to remind you that we encourage the sponsor to request a pre-CTA meeting to ask questions and advice when you feel ready. And these meetings are free.
And similarly, if you have any questions about GMP at any point, please contact Drug GMP Questions at our email. There is a 30-day service standard for responses. And just highlighting that the Office of Clinical Trials is also available to answer any questions about clinical trials.
And finally, we would like to thank our colleagues for their help provided to prepare this presentation. Thank you.
Thank you.
That was terrific. Thank you very much. Wow, that's a lot of content we've gotten this afternoon. We are going to move into the last phase of our webinar today, and I will ask those who have presented to come online, and our three speakers will also be joined by two new additions, Gail Payette and Dr. Jennifer Quizzi. Gail has a background in medical lab technology and over 30 years experience working in the life sciences field, with 12 spent specializing in good laboratory practices and another 12 spent in good manufacturing practice. In 2011, Gail joined the University of Alberta as a project manager for the construction of a GMP cell and gene therapy facility, and is now the director of the Alberta Cell Therapy Manufacturing Center there. Dr. Jennifer Quizzi has a doctorate in cellular and molecular medicine from the University of Ottawa, and has been working as part of a translational team in cancer therapy, providing strategic and scientific insight for more than 10 decades. As Director of Manufacturing Operations at BioCanRx, Dr. Quizzi is responsible for enabling the rollout of point-of-care, or POC, manufacturing at strategic sites across Canada, as well as facilitating the building of additional capacity in therapeutic virus manufacturing in Canada. She's also an investigator and the director of BioCanRx's core facility, called the Biotherapeutics Manufacturing Center, Virus Manufacturing Facility at the Ottawa Hospital Research Institute. So really wonderful to have Gail and Jennifer join our other panelists. for our Q&A session. I have, I think, about 5 different questions in the Q&A box. And so I'm going to just take us through those, as I know we're limited on time today. So the first question that was asked, Brian, I think this one's for you. Do all reagents at the start of manufacturing need to be GMP grade? And there's an example used for plasmids used for viral vector production.
As we just heard, there's a definition of start of manufacturing. So I think it's true that once you start manufacturing, you are in the realm of GMP. When we if we are making a new, let's say an adenoviral vector vaccine, we don't use GMP grade plasmid to rescue that virus. We rescue our viruses on very well characterized HEC293 cells in our facility. And we document the heck out of everything we do to do the rescue. And then we do several rounds of plaque purification, because it's very important to have an actual clone that you pass off. I think it's fair to say up to that point, you don't have to actually be GMP, but the truth is the better you document everything, up to that point, so that what you hand to a manufacturing facility is very well defined, the better off you are, because you may get questions about, where did this come from, and how were the cells that it was rescued and grown, or where did the plasmids come from. The more you can document, the easier it is to answer those questions later.
Can I maybe add something to there from the gene therapy perspective as well? So I think that this is an interesting conversation in that space, and it really depends too on what your final product is. So for instance, using, let's say, a lentivirus to make a CAR T cell, we've encountered this question quite a bit. And I also would love to throw this to my colleagues at Health Canada, because it's been through our engagement with Health Canada that we've been able to decide on our route forward. It usually starts off with doing a risk assessment to figure out what the potential impact could be of using plasmids in the case of gene therapy vector that are not of GMP origin. So for instance, for a CAR T, as I was mentioning, where we have the plasmid being used to make the lentivirus, and then the lentivirus in turn is used to make the CAR T product, that has been determined if you have a GMP source, so not under GMP conditions, but that we have documentation to show that the plasmid is, clean, so sterility, endo, myco have been done, that would be suitable. Very much to Brian's point that, documentation is a key component to being able to de-risk that. I think where there's still a bit of a question, and I think a conversation to be had for these early phase clinical materials that we're generating are more in the space of like an adeno-associated virus, where, when we're generating that vector, it is that vector that's then going to be administered to a patient. So perhaps the source of the plasmid is more important in that sense. And so this is where, again, we've had ongoing conversations with Health Canada to determine the suitability of use of some of these reagents, depending on what the product is that's ultimately administered to the patient.
Okay, thanks very much for that answer. The next question. I've heard that a potency assay is now required for phase 1 trials. Marie-Michelle's slide suggested that is only required for later phase. Could you elaborate on the requirements, please?
Potency assay, it's needed for phase 1 at the beginning. On the slide is indicated a qualified potency assay, and I said like a robust potency assay. So at the end, in phase 3, we expect like a strong, robust potency assay, but like at the beginning, a potency assay is expected, but maybe it's not like the qualified, the one that would be used for all the clinical trial, but for sure, at the beginning, we need a potency assay.
Although I think I could add here, even though it's not entirely my realm, potency is a moving target, I think, right? So for a vaccine, your potency assay could realistically be the titer or a TCID50 or some measure of how many active viral particles are in a viral vector vaccine. I would imagine that the potency assay for a CAR T-cell product would be a very much more difficult thing to assess. So is it true that potency assays need to be widely adjusted depending on the nature of the biologic?
Yeah. But then CSA is not a safety assay. So for sure that at the beginning, phase 1, we focus on safety, and eventually you want that the efficacy, you want that your drug is working good and to show that it's working. But I understand that at the beginning, it can be hard to find the right assay of potency, but at least to show that something is going on for phase 1, it's the minimum.
And
I think
if
of it as actually, what is your dose? How do you know what is your dose?
I was just going to comment too that for our clients in cell therapy, often the potency assay is done by the sponsor and not transferred to us at GMP. And specification is often not set. There will be a for information only value given until more batches are done and more data has been generated.
Thank you for that. And so this is a question I've heard before. What's the right time to approach Health Canada for a pre-CTA meeting? And what information is required beforehand for the meeting to be fruitful?
Yeah, I think that when you have a good idea of your manufacturing process, your control, and then you can, like, as soon as possible, you can request a pre-CTA meeting. It's the waiting time, it's a little bit long also, that you can do it in advance. And so, and it's the time to ask questions that you're not sure about the safety, and how to proceed also with the filing and the regulatory. So that's I think that as soon as you know, like, how you want the manufacturing process and how you have a good idea, because you will need to submit to us a package 1 month in advance for the reviewer to go through and through the question. So like all the section I mentioned today, so you can provide some information about this section. Yeah.
Thanks. My own supplementary. Can you have more than one pre-CTA?
Yeah.
Okay. So I hope that's helpful, folks.
Maybe just on that note, though, too, maybe another extension of that question is when you engage with GMP manufacturing, because as Marie-Michelle just mentioned there, right, you do need to have information, albeit it can be somewhat rudimentary about the process, that you're going to use to manufacture, if that's in fact what you were looking to do. And so having engagement with someone like myself or Gail or Brian, one of our facilities, early on is going to be important, because that will be important information that you want to include in that pre-CTA package, and also with how regulations are evolving in basically real time, because biologics are changing quite rapidly. This is where it's super important that we have some really good understanding of those key process parameters that will be incorporated into your production run, because if we don't ask those questions from a production perspective early on, we then go into GMP and make your biologic. But if we haven't taken the appropriate retains for some, of the release tests, or have the right amount set aside for the stability, then your clinical program could be in jeopardy. So I would say adding one extra step there, you'd want to engage with the biomanufacturing folks prior to engage with Health Canada, because then you'll need that information from us to help have a fruitful conversation with Health Canada.
Great. Thanks, Jim, for that. I think that's really good advice. So how can a sponsor get advice on facility design in an academic setting for a phase 1/2 trial? Anyone have anything on that?
Well, I'm not sure exactly what the question's about. I mean, if the thinking is that you want to build your own manufacturing site, there's lots of expensive consultants out there who will help you design and build a facility.
Yeah.
What else to say? Yeah.
Yeah,
Okay.
just sorry, I just wanted to add something really quickly. Most of the CDMOs who will be doing the manufacturing would already be very well versed with the facility requirements. And like Brian mentioned, to create a facility from scratch requires a lot of effort. That being said, Health Canada has published a lot of extensive guidance on the expectations that facilities must meet, both in the expectations for their premises and their equipment and the validation master plan that you have to have for your facility. That's all there from Health Canada, and you can also find guidance published through resources such as ICH or through other compendials. USP is a really great resource for some supplementary information. I'm not sure if that answers your question, but if you have more questions aside from that, as Brian mentioned, there are also many consultants who are available to provide advice that's more tailored for your situation.
Yeah, and if I could just add to that too, I think it's important to understand that facilities will look different depending on what type of product that you're manufacturing. So whatever your advice that you get, you should make sure that the people are well versed in that end product.
Thank you. Is there a requirement to have an established quality management system before starting the process of translation and an R&D product to a GMP product?
And maybe on this, that the sponsor is responsible for to maintain all the record regarding the clinical trial. Also responsible for reporting a serious unexpected adverse drug reaction. And so all these you need to have a system in place to record this quality information.
Okay. And to move on from there, I'm so glad to see so many questions. How is potency in cellular therapy really determined or measured appropriately? And how do you apply a specification to something that's inevitably variable and patient-specific?
I think Gail mentioned, I think it was you, Gail, that especially for the early phase, for some of these activities, it's to report, because one, we don't know, and two, it could be hyper variable, especially considering that some of the starting material can be also obviously very, very different. It's patient dependent. As far as for later stage potency, I would defer to our colleagues here at Health Canada to provide some maybe examples of best to broach that. But I'd say in the space we operate, it's usually to report at this point in time. And we do look at the clinical outcomes of the patients afterwards. And I say that not as a clinician, but being part of programs that do have that extension, obviously, where we have line of sight to how the patients are doing.
Yeah, and I think I would add too, it's perfectly fine to have more than one assay. And you may learn more information by having additional assays added with time as well.
Yeah, I think having more than one assay, you based on the biology and your knowledge, you pick things you think should correlate with potency, but they're just to report, and you get the clinical data, and then you figure it out. Because over time, you are going to narrow things and set specs, but you don't need specifications for everything to begin with. And Health Canada can correct me if I'm wrong, but I'm pretty sure of that.
Yeah, that's correct. Report results are expected for some specification at early phases, and yeah, as long like there is a justification, and then normally in phase 3 CTA application, we expect a quantitative specification.
Okay, we have a bunch more questions. I know we're not going to get through them in 4 minutes time, so I am going to ask one last question before we wrap up. If we opt for RNA vectors in cell therapy, how does the manufacturing process differ from using viral vectors in the context of biologic GMP manufacturing? Specifically, what are the unique considerations or challenges associated with RNA vectors in this manufacturing process? We're thinking hard.
Well, my short answer, unfortunately, is I don't know. I've never made RNA vectors, so I don't know if the others can comment.
Jen, do you have any expertise in that area?
No, I was going to say the same thing. I mean, yeah, we don't manufacture RNA, so can't really say, but I would assume, you know, the differences, I don't know that they're terribly important. You'd have to create a process obviously specific to the product that you're making. Could there be advantages? Sure. I'm assuming in the amount of testing. I mean, the fact that we're making viruses means that we have different assays, because part of what Health Canada was even describing is that as one of the release criteria, trying to make sure there's no viruses in your product. Of course, our product is a virus. So, you know, there's just different, I guess, requirements. But because I don't make the RNA, I don't make GMP RNA, I can't really comment on that specifically.
Okay, well, we've got 2 minutes left, so I'm going to combine two questions here that we've got. There's a question here about procedure and requirements for a pre-CTA meeting and what individuals on the applicant side should attend that meeting. There's also a question about, is there a drawback to requesting a pre-CTA meeting too early, and how that might impact future interactions with Health Canada?
So yeah, for the first question, how to proceed. On the slide, I put the email to contact the ORA, the person, like you can just contact them and they will organize everything. And for the impact for future interaction, like you can ask question, you can draw back. There is like no impact for the actual CTA authorization.
Okay, and on that, we are 1 minute before our end. We are very prompt and efficient with our time. I'd like to thank all of our guests today who've participated in this session. I hope our audience members who are here, which there was over 100 of you at one point, enjoyed this webinar and were able to get out of it some valuable information. I'd note that the next webinar takes place next Monday, November the 27th at the same time. It will be brought to you by our friends at BiocanRx, and it's called CMC Strategy, Supporting Cell and Gene Therapy Translation from Concept to Clinical Products and Beyond. You can find more information and registration on Biocan's website on the training page. And so if you haven't signed up for that, please feel free to do that. And as a reminder, this session and following sessions have been recorded. They'll be posted to Stem Cell Network's YouTube channel, and that's where you'll be able to access the slides that were presented today. So thanks very much, everybody, for joining us, and have a good afternoon.